right, so hi everyone. Thank you so much for joining our early career researcher event today, which is on the gut microbiome and its role in cardiovascular health and disease. We have a really great lineup of speakers today who are here to share their work with us. But before I pass it off to the speakers, I just want to share a few company slides to introduce Proteintech to you. So Proteintech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that cover many different research areas. We also offer antibody labeling kits, ELISA kits, IHC kits, and cell separation kits. We also have our humankind branded cytokines and growth factors, and all our proteins are expressed in human HEC293 cells so that you can be confident that they have native folding, post-translational modifications, and glycosylation. We also have our Chromotech branded nanobody-based reagents, which are based off of alpaca nanobodies. And because of their small size, they are ideal for immunoprecipitation, immunofluorescence, and protein purification. I also just want to take a minute to celebrate the success of our customers because our customer success is also our success. And we're really proud that Proteintech products have been cited over 200,000 times worldwide. And we're also really excited that we were the winners of the Researcher's Choice Award last year from SiteAb. So with that, I just want to introduce our speakers for today. So we have our keynote speaker, Dr. Jeff Salomon from the University of Nebraska Medical Center. We have Dr. Monika Gawalko from the University of Warsaw, Emerson Nolasco from University of Nebraska-Lincoln, Dr. Prasenjit Saha from Cleveland Clinic, and Sachin Aryal from University of Toledo. So I want to first introduce our keynote speaker for today, Dr. Jeff Salomon. So Dr. Salomon is a pediatric critical care physician and assistant professor at the University of Nebraska Medical Center in the Department of Pediatrics and Cellular and Integrative Physiology. He earned his medical degree from Saba University School of Medicine. He performed his pediatric residency at Driscoll Children's Hospital in Corpus Christi, Texas, and his fellowship in pediatric critical care medicine at Cardinal Glennon Children's Hospital in St. Louis, Missouri. His research focuses on how the microbiome influences inflammatory signaling after pediatric cardiac surgery and outcomes. He is currently funded by the American Heart Association and the Child Health Research Institute at UNMC. So with that, I will go ahead and pass it off to Dr. Salomon. Thank you so much for being here today and for sharing your work with us. If at any point you guys have questions for Dr. Salomon, please input those in the chat in the Q&A session, and we will address them at the end.
All right, well, thank you so much for that warm introduction. I'm going to go ahead and share my screen here. And we will start this presentation. Can everyone see my screen okay?
Yes.
Perfect. Well, I just want to start and say thank you for the opportunity to present today. My research, as I'm probably a little biased, is an area that's well needed in pediatric congenital heart disease. And not many people are focusing on this area of research in pediatric heart disease. Since this is an early career researcher event, I wanted to take just a minute and highlight a little bit about myself because I did not have a very traditional path into research. And so there may be other individuals that are sharing a similar path as mine in this unique format. And so I did not get into research until my fellowship. I really wasn't in a lab, had no basic science experience. And I attended a talk at the Society for Critical Care Medicine conference in 2017 that talked about the pathobiome in critical illness. And I noted a lot of similarities between critical illness and patients after they have cardiac surgery with bypass, and the low cardiac output, the inflammatory milieu, and the phenotype being very similar. And so I asked the question, does the microbiome changes that occur in other sepsis and critical illness occur after these patients have cardiac surgery? And I was directed to partner with a GI physician who had a lab and we developed a prospective study from scratch that was published in JACC BTS. But that really kind of made me do a 180-degree career change into research. If you had asked me when I started fellowship if I was going to be a researcher, I would have laughed in your face and told you no way. And so just wanted to share that, not everyone's path is going to, be the same the whole way. Be open to those opportunities that may take you down a different road, and know that there are going to be many ways to achieve success in research and get that experience and progress. So I just wanted to, I know we're limited on time, but wanted to just kind of share a little bit of that background about me, and happy to answer any questions later in that regard as well. So my talk is going to be focusing on the relationship of the microbiome and intestinal milieu associated with congenital heart disease. And so I have no disclosures other than where my research funding comes from. And the objectives of this talk are to distinguish the different components of the intestinal microbiome, identify the significance of the intestinal barrier and some intestinal metabolites, understand the characteristics of the microbiome on heart disease and congenital heart disease, and then recognize how the microbiome, barrier dysfunction, and the metabolites can contribute to inflammation after cardiopulmonary bypass. So briefly, congenital heart disease affects roughly 40,000 children every year, and roughly 25% of those will need cardiac surgery within the first year of life. Now, mainly congenital heart disease has two types of classifications: cyanotic with low oxygen levels, or acyanotic. And then those can be further divided, whether there's increased pulmonary flow or decreased pulmonary flow, whether we have left to right shunt lesions on the acyanotic, or we have certain types of obstructive lesions. And so while we have made significant strides in the last few decades in improving the outcomes after these pediatric patients undergo heart disease, it is still one of the leading causes of death in under 2 years of life. So cardiac surgery and bypass in general congenital heart disease affects gut perfusion. And this can either be with patients when they have hypoxemia or low oxygen levels, or whether they have reduced systemic perfusion, which can be broken down either to a low cardiac output state, or whether there's increased pulmonary overcirculation, where too much blood is going to the lungs and not enough going to the body. The cardiac surgery to repair the majority of these defects will require the use of cardiopulmonary bypass. Now, we know this bypass induces a systemic inflammatory response syndrome. This leads to increased cytokine and inflammatory mediator production, increased intestinal permeability, and in some cases, especially in neonates and our young infants, can lead to a low cardiac output state. Now, switching gears into the microbiome, we well know that it is important for health and homeostasis. It is composed of over 100 trillion bacteria, viruses, and fungi. So there are more bacteria in your gut than there are cells in your body. Now, the 5 most prevalent phyla are Firmicutes, Bacteroidetes, Actinobacteria, and Proteobacteria, along with Fusobacteria. Now, recently, there's been a taxonomic change in how these are described. So Proteobacteria does not have the same name. Bacteroidetes does not have the same name. And so as you see future manuscript production publications, that's going to be changing. And so those names are going to be changing. The 2 most common phyla are Bacteroidetes and Firmicutes, and these make up nearly 90% of all species in the gut. But many pathogenic organisms occur within the phylum Proteobacteria. As we well know, these cause disease: E. coli, Salmonella, Shigella, Yersinia, Pseudomonas, and many others. Now, the microbiome is not static. We also know that this changes significantly over time. The bacteria that live in your gut that you are born with is based mainly upon the mode of delivery that baby had. So if you had a vaginal delivery, then vaginal flora is what colonizes the gut early on. If it was a cesarean, you have skin flora that colonizes the gut early on. And so these populations will shift over time. So just before puberty, so about 7 years to 10 to 11, there's a little bit of a plateau. We have another shift with puberty, and then it does kind of stabilize into adulthood, but then shifts again into old age back to a makeup of your microbiome that was actually relatively similar to what you had as an infant. And so when we talk about the microbiome, there are some definitions to keep in mind that are important. So alpha diversity, which is the observed richness or the number of organisms, or evenness, the relative abundance or percent of these different organisms in one specific ecosystem. Then beta diversity is how the change in species between two ecosystems or an ecosystem at different time points can vary. So again, the other important thing that goes along with the microbiome in the gut is something we call the intestinal barrier. And so this is made up of multiple layers. So we have the mucus layer that exists in the lumen of the gut and is on the villi of the epithelial cells. And so there's a barrier there, but then we also have tight junction proteins. And so many of these are these claudin family of proteins, and two very important ones are claudin 2 and claudin 3 in the gut. We also have occludins, which can be found in endothelial cells and along many of our epithelial borders. And so when there's dysfunction, we have this cellular membrane disruption. So many of these tight junctions are wrapped around both sides of two cells and keep them together. But many of them also act as pores or gateways to allow things of different size or charge into through the epithelial line from the lumen into the deeper levels. So when there's cell damage or disruption of this barrier, then there becomes unrestricted pathway for the bacteria, the toxins, and these metabolites that can leak out of the gut lumen into the deeper tissues, which can have a lot of downstream signaling. So one of the things we've started to identify that we can help note when there is this dysfunction is when we have increased intestinal fatty acid binding protein. So there's a family of these organisms, but FABP2 is an intestinal specific one, and it is an intercellular lipid transporter. And so when you can find this and measure this in the plasma, that means that there's been cellular damage and these are leaking out. We also can measure the claudin proteins in the plasma as well. And so when we can measure these, that kind of equates to gut leakiness or this barrier dysfunction. And so that gut leakiness is that increased permeability that I mentioned, where more things that are supposed to stay in the gut lumen are allowed to leak into the deeper tissues of the gut. Again, that can be bacteria, toxins, and metabolites. So how does this relate to heart disease? Well, many things can affect this, the microbial composition as well as the function of the barrier. And so one thing that we've learned over time is that the Western diet, not so great. But it has less fiber, and so that leads to less short-chain fatty acid production. And it also leads to increased trimethylamine from choline and carnitine conversion. So then ultimately, what we see here is that butyrate and other short-chain fatty acids that are important to maintaining that intestinal barrier are also found to be cardioprotective. And so when there's abnormal microbial makeup in the gut, what we have found is that the organisms that are responsible for making those short-chain fatty acids are reduced in number. And so when there's less of those, there's less signaling for the barrier to maintain its integrity. Additionally, TMA then goes to the liver and it gets converted into trimethylamine oxide. And this has been associated with foam cell formation, cholesterol transportation disruption, and increased platelet activity. And all of these go on to develop atherosclerosis, which can lead to acute coronary syndrome and ultimately heart failure. And so then another thing dealing more specifically with the short-chain fatty acids are these postbiotics and the downstream effects. So we are learning more about how important these short-chain fatty acid molecules are. And so what we found is that short-chain fatty acids can stimulate prostaglandins, and these can increase signaling to MUC2 genes, which increase the mucus production and barrier. We've also found that there are certain fatty acid receptors and intestinal olfactory receptors that can activate the jugular glomerular apparatus to modulate blood pressure and heart rate. And then we've also found that these inhibit production and activation of histone deacetylases, which actually reduce the amount of inflammatory signaling. And this can modulate the epigenome as well as modulate immunity, such as reducing the activation of hypoxia-inducible factor 1 alpha and nuclear factor kappa B inflammatory transcriptional pathways. This also can reduce the amount of neutrophil chemotaxis and increase the amount of T regulocyte activity to, again, further downstream modulate that inflammatory signaling. Of note, not only do these work on the gut and other systemic effects, short-chain fatty acids in general have been found to be heart protective in and of themselves. And so there was a study in this ischemia reperfusion model where just direct administration of short-chain fatty acids into the abdomen, not the gut, not the bloodstream, just right into the peritoneum. And they did this ischemia reperfusion on these rats, and they found that propionate and butyrate, two of the most important short-chain fatty acids, were noted to visually reduce the infarct area in these ischemia reperfusion modeled rats. And then histologically, they found that both the area at risk and the infarct area itself was reduced with propionate and butyrate when they looked under the microscope at the amount of inflammatory infiltration in histologic samples. So short-chain fatty acids can do a lot to have many systemic effects, but are directly cardioprotective. And then specifically in congenital heart disease, there has not been a lot of research in this area. One good study that came out looked at the microbiome in congenital heart disease. And so we had 50 patients in each group. And what's of note here is that the 2/3 of these patients had aortic stenosis or atresia or pulmonary stenosis or atresia, which means that the majority of these patients essentially had a single ventricle type physiology. And so that becomes important because these patients either received a placebo or a symbiotic with bifidobacterium bacteria, inulin, and mayflower daily from enrollment until discharge or death. And what we were able to find was that the patients in the symbiotic group had a statistically reduced amount of time requiring mechanical ventilation, diagnosis of NEC at least or equal to grade 2, and then sepsis, clinical and proven sepsis were reduced in the symbiotic group compared to the placebo group. And then there was a reduced mortality in the symbiotic group versus the placebo group. Now, this was only one study, and it kind of highlights that the lack of information, but also the amount of information that we still need to learn about these patients. And so what we're really missing is an understanding of the microbial makeup in these patients. And then what role does barrier function play in this process? And then how can we properly evaluate the microbiome and barrier function? And so that's where my research kind of really got focused. And so I developed a study where we started looking at some control patients that did not have heart disease, but were undergoing similar surgeries as far as the duration, the anesthetics that were going to be used, and the intraoperative antibiotics, and at least going to be in the hospital 2 to 3 days after their surgery. And then our experimental group was our patients with congenital heart disease undergoing cardiac surgery with bypass. And so what we can see here is that on the top, we have our control patients. The post, unfortunately, is on the left and the pre is on the right. But what you can see is that at baseline, this gold is these proteobacteria, so many of these pro-inflammatory organisms. And so at baseline, the patients with congenital heart disease had significantly higher amounts of proteobacteria than our control population. And it's pretty easy to see that this was markedly increased after surgery with bypass. And so the bypass was just further propagating the production of these pro-inflammatory organisms. Now, getting into alpha diversity, which I spoke about previously, there are multiple types. So the Shannon, these are just 3 different indices, mainly focusing on evenness or the distribution of different organisms. And so again, we have control pre and post on the left, and then our bypass group pre and post on the right. And so what we can see is that there was a reduction in the alpha diversity, or the number of different organisms that were present in these patients after they underwent cardiac surgery with bypass, while our control group really did not experience a significant reduction in the amount of organisms after their surgery. Specifically looking at different phyla, we can see 3 ones of import are Firmicutes. So there was a significant reduction in the amount of Firmicutes organisms in our bypass population. And there was a statistically significant increase in Proteobacteriae and Varicomicrobiota after cardiac surgery with bypass in our experimental group. So looking at the genus level, we can see here, so this is a waterfall plot. So this is comparing our bypass group post to the control group. And so what we can see, the red is saying organisms that were increased in abundance. And then the blue-green is what were decreased in abundance. And so what I've starred here on the bottom are genera that are associated with short-chain fatty acid production. And so we can see here that there are quite a few different groups of organisms that produce those short-chain fatty acids that were reduced in our bypass population after. So then we wanted to know what was going on with the intestinal barrier. And so again, we looked at claudin 2, claudin 3, and fatty acid binding protein 2. And so what we can see is that all of these were statistically increased after cardiopulmonary bypass compared to our control group. And again, my control group were neurosurgical patients and spinal fusion patients. So these are still big operations that have the potential to cause inflammatory surges postoperatively. Then we also wanted to know what was going on with the cytokines. So again, getting into kind of the inflammatory side of what's happening. While cytokines are still kind of viewed as a crude measure of what's happening, it's one of the few ways that we can kind of measure these byproducts of inflammation. And so what we found is that at 1 hour post-op, after they return to the cardiac unit, we can see tumor necrosis factor alpha, interleukin-1 beta, IL-6, and then IL-8 were all statistically increased in our bypass patients compared to the control patients. Now, this is not a surprising finding, right? We all know that bypass in and of itself causes an inflammatory surge, as I've already discussed. But it was just of note that our control population really did not experience any significant increase in cytokine production. And so then we wanted to know what was happening specifically with the human short-chain fatty acids. And so what we saw was that there was a statistical reduction of acetic acid, butyric acid, and propionic acid in our bypass patients compared to our control patients pre-op to post-op in the stool samples. And so we did measure these in plasma as well. I haven't included that data, but those were also decreased in butyric acid and propionic acid as well. But this is kind of showing us that, again, because short-chain fatty acids are made almost exclusively by organisms in the gut, that because the organisms that make them were decreased in number, that then the short-chain fatty acids themselves would be decreased. And so here is just looking at what we call a pie crust analysis. And so this is looking at metabolite pathways based on the bacterial DNA that we got from our sequencing information. And so what we can see here is that some of these pathways, the phenylacetate degradation, the hydroxyphenylacetate, and the phenylpropionate degradation are all involved in the pathway to reduce the amount of downstream short-chain fatty acids, or your final short-chain fatty acid metabolite. Interestingly, we also found, so this LPS biosynthesis and then some antibiotic resistance pathways were increased in abundance. And so as we're now shifting to go from 16S data into whole genome sequencing data, it'll be really interesting to see how we can better map out these metabolite pathways once we have more detailed data. But this was just kind of a snapshot and what kind of perked our interest that we can look at later as some metabolite pathways that may be targets downstream. So then I also wanted to know, because it's kind of harder to do, get approval to do studies and interventions on pediatric patients, were there things that we could also identify similarly in a piglet bypass? And so one of my mentors at the University of Colorado, Dr. Jesse Davidson, has a piglet model of bypass where they undergo deep hypothermic circulatory arrest, which is something that's done for a lot of aortic arch repairs and different complex left-sided heart lesion repairs. And so we put these piglets on essentially more like an ECMO circuit than a true bypass circuit, but we cool them down to 18 degrees and then had them on deep hypothermic circulatory arrest for 100 minutes. And then they were supported for 6 hours after and then euthanized. And the control group underwent mechanical ventilatory support for that same duration that the piglets had DHCA and bypass support. And so then these stool samples were collected right before intervention and then at euthanasia in both groups. And what we were able to see was, again, because the time frame was so short, there wasn't as quite a significant reduction, but in the control post versus the bypass post pigs, we did see, a statistically significant reduction in phylogenetic diversity. And phylogenetic diversity is a little unique in the way as it looks at the evolutionary distance of organisms on a taxonomic tree. And so the further they are away from each other, the more phylogenetic diversity you have. And so you had, what this shows is that there was kind of a narrowing of the type of organisms that were in the gut on the evolutionary timeline. And then this graph here on the left in the C panel looks at this beta diversity. And so while it's a little challenging to understand, what you can see is that we had the pre and post time points in each group and how the overarching shift of these organisms. And so what we found was that there was statistically significant beta diversity shift in between these two ecosystems and these populations. So we also wanted to look a little bit more at some of the other metabolite profiles. And so again, we're still trying to tabulate all the data in our humans right now. But in these piglets, we were able to look at some of the metabolite profiles. And what we were able to find is that post-operatively, the control piglets, which are in the red box, were uniquely separated than the blue box. And so there were these metabolite profile shifts that were unique to the piglets that underwent bypass. And so that'll be interesting as we kind of determine what kind of mechanistic targets we can look at in the future. We also wanted to know, can we do any mediation analysis? Was the microbiome itself actually responsible for any of these changes? And so what we found is that the microbiome was responsible for at least a statistically significant portion of the change in prostaglandin D2, prostaglandin E2, and valeric acid. So I was also thought we might see a unique mediation for our barrier dysfunction markers, our claudins and FABP2. Unfortunately, those did not pan out. And so my personal hypothesis right now is that the bypass itself induces the barrier dysfunction, and then the existing dysbiosis and altered metabolite profiles are allowed to leak out of the gut and influence further downstream inflammation. And so that's kind of our bottom line, right? So we have this dysbiosis and these altered metabolite profiles after cardiac surgery with bypass. We found that bypass can exacerbate these differences, and that bypass does induce this intestinal barrier dysfunction that leads to some other downstream effects because of the metabolite profile and microbiome that exists in these patients with congenital heart disease. And then we did find that, at least in the piglets, we're still waiting to do this mediation analysis on my human samples, that the microbiome does have the ability to mediate changes in inflammatory metabolites and is responsible for reductions in some specific short-chain fatty acids, which all are noted to have downstream effects on inflammatory signaling. So my ongoing areas of research are really kind of looking at a few different angles. So one of them is, again, noticing this hypoxemia and low cardiac output that we can have in patients with congenital heart disease. And based on the microbial changes that we've already identified, how is that signaling downstream immune cells? So we're performing flow cytometry and immunofluorescence to kind of get better understandings of the breakdown of these different cell populations. And then we're also wanting to do RNA sequencing. So we have an RNA hybridization panel that's looking at about 200 inflammatory genes on these isolated immune cells. And we are collecting these samples before and after cardiac surgery. And so we can see kind of what inflammatory signaling and expression is happening with certain inflammatory genes. And then how is that going to be then correlated to the production of pro-inflammatory cytokines? And then on the opposite side, we're working on getting our own piglet bypass model up and running here at UNMC, where we can then actually do tissue samples, where we can look at DAMPs and PAMPs on the intestinal epithelial cells based on the microbiome and pro-inflammatory organisms that exist. And then how these pattern recognition receptors respond, and then what kind of activation in the epithelial cells for some of these transcriptional inflammatory pathways. And then how that, again, leads to increased production of cytokines. It should also be noted that some of these actually end up being host protective responses, right? So we have kind of a balance in this inflammatory signaling. So we have anti-apoptosis, antioxidant properties, and antimicrobial peptides that can be produced by different cells in the body. And so it'll be interesting to kind of identify these with what's happening with microbial shifts and how there may be a balancing act in some patients that we may be able to upregulate to temper some of the inflammatory cascade that happens after bypass. So just recently, we started to get our first panel back of these inflammatory gene RNA hybridization panels. And so this was looking specifically at 5 different patients combined with single ventricle physiology, so full mixing lesions that underwent their first stage palliation repair. And so whoops, I'm sorry. So on the first panel here on the left, again, I'm still working with the software to figure out how we get everything labeled correctly. But we have our pre-op samples and then these 1 hour post. And it's pretty interesting that there's a lot of these inflammatory genes that almost flip-flop in their activation. And so we can see CXCL2, which are chemotaxis genes, NOD1, same, CXCL9. And then we have our hypoxia-inducible factor 1 alpha, which was interesting because I actually thought this would have been upregulated given that these patients are hypoxemic at baseline, but it was turned off and then had an increased signaling post-bypass. And then we had nuclear factor kappa B, which was upregulated and then turned down in expression in the 1 hour post. So it'll be kind of interesting as we get more patients and can kind of look at this data in a little bit more detail. And then on the right panel, again, post-op day 3 is kind of in the second column. Again, I don't know why it got put there, I'm trying to sort that out, but kind of worked out nicely because what we can see is that by post-op day 3, a lot of those genes that were switched in the early post-operative period are actually going back to similar preoperative expression patterns. And so what is inducing some of those things and some of these changes to flip them on and off will be really interesting to look at when we have more data and we have our animal model and can look at this in more detail. And so again, more specifically here with some of these specific inflammatory genes, we can see that there was a statistically significant reduction in expression from the pre-op to 1 hour post-op with nuclear capture factor B. And then we had increases in the HIF1 alpha, interleukin 1B, and CXCL2 gene activations. And so again, as we get more patients and have more of these panels and data to compile, it'll be interesting to see how that plays out, especially with different cardiac defects and different surgeries that are performed, even potentially related to the duration of bypass, cross-clamp, and some other things that happen intraoperatively. And so I'd just like to take a moment to thank my current mentors, Dr. Amar Singh here at UNMC and Dr. Jesse Davidson at the University of Colorado, some people that have worked in my lab, a medical student, Jacob Owens, and my 2 research techs. And then I have my scientific advisory team, the Child Health Research Institute and the Department of Pediatrics for getting me started on this journey here. Some individuals at the University of Nebraska-Lincoln who have helped me with all of my microbiome bioinformatics. And then the lab here at UNMC that helps me with my metabolomic profiling. And again, just the hospital and UNMC system for giving me these opportunities. And always above is my beautiful family, wife and daughter, who I do all of this for. And I'd like to thank everyone for this opportunity to share with you a little bit about the microbiome and congenital heart disease, my research, and what we found so far and what we hope to find in the future. And I'll open it up to any questions.
Great. Thank you so much, Dr. Salomon. That was really interesting. We do have some questions in the chat and the Q&A. So the first question is, does the normal flora change from person to person?
Yes. So there are many factors that change the normal flora. Your diet. In infants, we can see breastfed infants versus formula-fed infants have different microbial makeups. But also, did you spend time in the NICU? Do you have any other baseline genetic disorders or things? There are so many genetic and environmental factors that make up the microbial distribution for patient-to-patient variability. The nice thing about the way I've designed my study is that each patient gets to act as their own control because we collect preoperative and postoperative stool samples, so we can account for a lot of that person-to-person variability.
Great. The next question is, can extreme intestinal membrane dysfunction cause septic shock?
So that's a great question. And I think the answer is yes. I think it has to be a fair amount of it, but one population that I feel like we've been able to identify this in is our premature babies. So they have very fragile guts. And it's not uncommon that we see gut barrier dysfunction that progresses into neck. And we see a lot of translocation of bacteria from the gut that causes a lot of those septic states. So yes, those premature babies can be more prone to marked perturbations of their intestinal barrier, which can lead to a lot of downstream diseases.
Our next question says, stool sample short-chain fatty acids are much higher in CPB than control. Why is this? Post-op samples have reduced levels, but these are now similar to control.
Yeah, so I don't have a really great explanation for that just yet. And then what does that mean on kind of the, barrier modulation? I don't know. One theory is that because there may be kind of this subtle, ongoing, like indolent inflammatory process, is the microbiome attempting to augment or modulate itself to be more protective by trying to increase some of those organisms that can increase short-chain fatty acid production at baseline. I think it'd be really interesting if I can get a few more samples pre-operatively and follow that a little more longitudinally in the pre-operative state in patients to kind of tease that out a little bit further. But that's a great thing that you identified. Yeah.
So this is an interesting question that I feel like we tend to have when we're working with model systems. Someone's asking, how does the pig and human gut microbiome compare to each other?
Great question. So a lot of people have asked me, hey, why don't you just use mice or something like that? Well, first off, putting a mouse on bypass is incredibly challenging. You're really using like 18-gauge angiocaths as your ECMO cannula. But in general, the pig microbiome is actually more similar than mice or rats to humans. Now we're talking, again, probably 96 to 98%. But when you actually get into the species and subspecies, we actually see a lot more similarities in the pigs to humans than we do in the rats or mice to humans. And so when you're wanting to do these models to really mimic a surgery or things that happen in humans, the piglets end up being a much better longitudinal model for that. Additionally, the tissue of pigs is much more similar to humans than mice. And so when we look at the barrier function in the intestines, it's much more similar than in rats or mice.
The next question is asking, did you measure gene activation through fecal or serum samples?
So we did serum samples. So we isolated immune cells and did an RNA hybridization plate with nanostring to identify the inflammatory genes. You raise a great point, and we're going to be looking at that in our animal model to see if we can identify inflammatory gene activation on the intestinal epithelial cells. I don't know of a good way to do that on like stool samples at this point, but when we have intestinal tissue samples that we can look at, it'll be really interesting to see kind of what's happening locally on inflammatory gene activation.
Okay, and I think we have time for one more question. If Dr. Salomon doesn't mind, he can type out answers after while the other speakers are speaking, or I can provide you guys with his email if you want to reach out to him personally and ask your question. But the last question that we will address is, does the composition abundance of the microbiome stabilize after a certain time point / age in humans and or animal models commonly used in research?
So that's a great question. The animal side, I'm not entirely certain for, the microbiome stabilization. I'm using infant pigs to try to mimic the infant state that most of these cardiac surgeries occur in. And so during, especially the first few years of life, there is significant ongoing shifts of the microbiome. Now, the time frame that we're looking at collecting this data is probably not going to have a significant, shift over a week period of time. But had we continued any animal modeling out longitudinally over months, it would not surprise me to see those shifts. The most aggressive shifts that occur are infants in the first month of life as they kind of transition to taking formula or breast milk from the delivery mode flora that dominates their gut in the typically the first week of life.
Great. Well, thank you so much again, Dr. Salomon, for your presentation, for answering all those questions. We will move on to our next speaker. This is the first of our series of ECR speakers that we have. This is Dr. Monika Gawalko. So Dr. Gawalko is a cardiologist in training at the Medical University of Warsaw. She underwent clinical and scientific fellowships at Maastricht University Medical Center, University of Copenhagen, and University of Duisburg-Essen. She is the author of many peer-reviewed publications in the field of atrial fibrillation, and her work has received several awards. Her research focuses on 1, mobile health solutions in atrial fibrillation, and 2, the mechanisms of atrial fibrillation, mainly dealing with the paracrine effect of the pericardial adipose tissue, and NLRP3 inflammasome, and gut microbiome. So with that, I would like to pass it off to Dr. Gawalko. Thank you so much for being here and for sharing your work.
Thank you very much for this kind introduction and for the opportunity to present this topic. And according to previous presentation, as we know, like a lot of most cardiovascular diseases have been shown to be associated with the gut microbiome, starting from the hypertension to the heart failure, coronary artery disease. And finally, the topic that I'm going to talk about is atrial fibrillation, the most sustained cardiac arrhythmia. So it's like only 72 publications on this topic, and half of them are just reviews. So it's like really unresearched topic. So before presenting some available data on the gut microbiome in the atrial fibrillation, I just want to briefly summarize that in the healthy status, the Firmicutes and Bacteroides contribute to the total bacteria species, and the ratio is almost 1. The ratio depends on the lifestyle components, and unhealthy diet, lack of the physical activity, medications can lead to the higher Firmicutes to the Bacteroides ratio and disease. This state can be reversed by approaching the healthy lifestyle attitude, as we can see on this really shortcoming figure. And in line, some small observational studies shown that different types of the atrial fibrillation as compared to the sinus shunt controls are associated with the gut dysbiosis and higher Firmicutes to the Bacteroides ratio. However, all these small observational studies haven't been designed to address the influence of the comorbidities and drugs on the changes in the gut microbiome and cannot demonstrate the effect-cause relationships. Also, big study like FinRisk 2002 that included almost 7,000 patients shown that although the atrial fibrillation was associated with this specific microbial genera differentiation, some partial overlap with the risk factors and the comorbidities were shown, what could influence overall assessment of the gut microbiome in the patients with the atrial fibrillation. Also, small studies have shown that some gut microbiome-derived metabolites, starting from the primary biliary acid, indoxysulfatate, lipopolysaccharide, trimethylamine, and oxidized short-chain fatty acids, and the choline, can influence the atrial fibrillation through different mechanisms, the inducibility of the atrial fibrillation. However, on all these intermediate lines, the data are not so yeah, how to say, it's not so safe, to be honest, and the most the best studies that relate the gut microbiota derived metabolites are focused on the lipopolysaccharide for the NLPV inflammasome. And before starting talking about the association between the lipopolysaccharide and the atrial fibrillation through the NLPV inflammasome signaling pathway, I just want to briefly introduce what the NLPV inflammasome is. So NLPV inflammasome is established signaling pathway responsible for the secretion of the inflammatory cytokines in the immune cells in the response of the cell damage and the infection. So the Yao et al. For the first time discovered the activity of the NLPV inflammasome in the atrial cardiomyocyte in the patients of the atrial fibrillation. And the cardiac myocytes activation of the NLPV inflammasome can enhance the atrial fibrillation through the calcium dysregulation, transcription of the potassium voltage-gated channels, and promotion of the fibrosis. And coming back to the enhanced atrial fibrillation through the lipopolysaccharide, in the study by Zhang et al., authors performed the fecal microbiota transcription from the aged rats to the young rats and young rats to themselves for the 6 weeks to investigate the role of the gut microbiome in the age-related atrial fibrillation. The aged microbiota recipient rats had the higher inducibility of the atrial fibrillation, longer atrial fibrillation duration, and also enhanced atrial fibrosis and upregulated NLPV inflammasome components as compared to the young microbiota recipient rats. To verify this stimulatory effect of the NLPV inflammasome changes, the authors administered the young microbiota recipient rats with the specific inhibitor of the NLPV inflammasome, MCC 950, and we can see that the inducibility of the atrial fibrillation was decreased. Also, decreased was the atrial fibrillation, and also when administered the inhibitor of the lipopolysaccharide, we can also see that the activation of the atrial NLPV inflammasome was also decreased, and also atrial fibrillation incidence and inducibility. And collectively, this data indicated that the lipopolysaccharide can enhance atrial NLPV inflammasome activity, resulting in the atrial structural remodeling due to fibrosis and subsequent atrial fibrillation. And coming to the another really common metabolites that was assessed in the patients with the atrial fibrillation, it was the short chain fatty acids. And it was like the first study that assessed like the role of the short chain fatty acid in the population of the patients with the atrial fibrillation. So the fecal, So the fecal levels of the short chain fatty acids were remarkably reduced in the patients with the atrial fibrillation, with declining from the paroxysmal to the persistent atrial fibrillation. The correlation between the short chain fatty acids and the left atrial diameter, which is the indicator of the atrial fibrosis and then marker of the atrial fibrillation, was negative, but without the significant, the statistical significance. And also in this study, to demonstrate the influence of the short chain fatty acids on the atrial fibrillation, authors fed the mice with the normal, low, and high fiber diet for the 4 weeks, showing the increased atrial fibrillation inducibility while consuming the low amounts of the fiber, hence short chain fatty acids. Also, in this study, the authors concluded that the role of the short chain fatty acid in the atrial fibrillation is based on the calcium dysregulation and also enhanced atrial fibrosis. And last metabolite that I would like to mention is that trimethylamine and oxidase is like the most extensively studied microbial-derived metabolite in the atrial fibrillation population. And in the Canning model, local injection of the trimethylamine and oxidase in the atrial autonomic ganglion plexus aggravated rapid atrial pacing, induced shortened effective refractory period reduction, and widened a window of the vulnerability, a surrogate of the atrial fibrillation inducibility, and all these mechanisms of the activation of the autonomic system through the trimethylamine and oxidase. And the pro-arrhythmic effect of the trimethylamine and oxidase was also confirmed in the clinical scenario in the meta-analysis of the 6 studies where the higher values of the trimethylamine and oxidase were shown to be increase the risk of the incidence of the atrial fibrillation. However, all these studies just assess once the trimethylamine and oxidase are at the baseline, and then assess the risk of the atrial fibrillation during like almost 10 years. And also another study that it's like really worth to mention for conducting further studies showed that when we are going to assess twice trimethylamine and oxidase, we are going to receive higher predictive value when we have the stable higher values than changes in the levels of the TRIMAO. So overall, repeated assessment of the trimethylamine and oxidase improve the interpretation of the people with the higher risk of disease endpoint. Also, in another 2 studies assessing the reliability of the different metabolites, trimethylamine and oxidase had the lowest reliability. So the low reliability of the trimethylamine and oxidase suggests that the single measurements is not enough. So it's really clear recommendation for all the further studies that to establish some specific gut microbiome derived metabolite as the biomarker is really crucial to assess that at least twice. So to conclude, gut microbiota, gut microbiome, can be important predictors and markers in the atrial fibrillation. However, more data are needed. The effects of the gut microbiome and associated metabolites on atrial fibrillation promoting mechanisms are poorly characterized. And if the gut microbiome modulates the atrial fibrillation and also other arrhythmias directly or indirectly should be also determined. And further investigation that, amongst other, I'm working on is the role of the pre, and post biotics on the atrial fibrillation. So how to use these medication supplements in the clinical scenario. Also, what is the difference between the individual and the whole bacterial communities and the impact on the atrial fibrillation and other cardiac diseases, and another non-bacterial microorganisms, because microbiome is not only bacteria, it's also viruses, it's all of fungi. So we have to just take into account another microorganisms. So thank you very much.
Thank you, Dr. Gawalko, for your amazing presentation. So if you guys have any questions, please do input them into the chat box or the Q&A section. While we're waiting for some questions to come in, I guess I have a question. So if, you know, a lot of times we see a lot of organ crosstalk and things like that. Is there evidence of, you know, gut microbiome, heart, and then brain interaction as well?
Yes, as I mentioned, like the most studied metabolysis was the trimethylamine and oxidase, and it was like really shown that if we are going to inject the ganglion plexus, it have like the stimulation of the autonomic system. So I think there is always connection. Whenever we are using one organ, it's like correlated with another. So that's why like the working on the gut microbiome is really hard, because always there is like the risk of bias that another organ is influencing our our results. And I think the best way is to first just focus on the animal models that are kind of clear model. It's not like the patients that almost, it's really tough to find the healthy controls without underlying diseases, and then established our results in the human model.
We have another question from Dr. Salomon asking, what was the atrial fibrosis change pre and post in the aged mice comparing with and without FMT?
So at the so this study just first showed that there was like the fibrosis in the old rats, of course, and the clue was to see if this fecal microbiota transplantation from the old rats can influence the fibrosis in the young ones. So it influenced that. So there was like the more markers of the fibrosis. So the young recipient rats developed the fibrosis. So.
Okay, thank you. And one more question before we move on is that only NLRP3 that's involved or other inflammasomes are also going up with AF?
So NLRP3 inflammasome is my main topic research, because I'm also working in the pericardial adipose tissue on the NLRP3 inflammasome. That's why I'm just only focused on this inflammasome, but also there are a lot another, I think almost 10 another inflammasome, but because the NLRP3 inflammasome is the most investigated one, we wanted to just establish one of them and then take another. So first one, but there are more.
Yeah, I mean, you can't do it all.
No.
Awesome. Okay, well, thank you so much, Dr. Gawalko, again for your talk.
Thank you.
We will move on to our next speaker, who is Emerson Nolasco. So Emerson is a PhD candidate at the University of Nebraska-Lincoln. He has expertise in assessing the bioaccessibility and bioactivity of food compounds using in vitro and in vivo models. His recent experience includes assessing the impact of digestion-resistant protein in the gut microbiome and its impact on human health. So with that, I will go ahead and pass it off to Emerson. Thank you so much for joining us.
Thank you for your kind introduction. Hello, everyone. Today, I'm going to talk about how the gut microbiome can modulate the anti-hypertensive activity of egg white hydrolysate. I'm going to start talking about cardiovascular diseases, which is one of the leading causes of mortality in the world. In these conditions, we can find heart failure and coronary artery disease, in which hypertension is a risk factor. Hypertension has different mechanisms which are often interconnected, but for the purpose of this study, I'm going to focus on 4: the oxidative stress, the vascular inflammation, the gut microbiome, and the renin-angiotensin system. There are already drugs that treat hypertension, but more often they're inaccessible for the low socioeconomic status population. They present side effects, and also they can only address one of hypertension mechanisms. And therefore, it's imperative to find natural alternatives that are cheaper, that have less side effects, and that can address more than one of hypertension mechanisms. One potential alternatives are food hydrolysates. Here, we take a food protein, and through processing techniques, we make a whole hydrolysate, which after passing through gastrointestinal digestion yields 2 fractions: a small molecular weight fraction, which is often more bioaccessible and have higher bioactivity, and a digestion-resistant fraction, where there is not much information out there in the literature, but I'm going to talk more about it in my next slide. In this study, we're using egg white hydrolysate because it has been shown to be a good source of anti-hypertensive peptides. Now, the status quo is that all of the protein that we ingest is hydrolyzed into amino acids and absorbed in our small intestine. But studies have shown that up to 5% of cooked and 30% of uncooked egg white protein remains undigested. And here we hypothesize that these digestion-resistant peptides can reach the colon, where the gut microbiome ferment those to produce new peptides or metabolites that could be detrimental to the host, such as ammonia and branched-chain fatty acids, or that can have positive effects in the host, such as the short-chain fatty acids, or that can even enhance the anti-hypertensive activity of the egg white hydrolysate. To test this hypothesis, we set two different objectives. The first one was to determine the egg white hydrolysate anti-hypertensive mechanism of action. And the second one was to characterize the egg white hydrolysate undigested fraction modulation of the gut microbiome and its metabolite production in vitro. We prepare our own in-house egg white hydrolysate through enzymatic hydrolysis. After we obtained the hydrolysate, we subjected it to a simulated gastrointestinal digestion, as well as egg white protein as a control. After we obtained the digesta, we separated into two fractions: the small molecular weight fraction, smaller than 3,000 Dalton, which was used to analyze the bioactivity of the hydrolysate, and the digestion resistant fraction, bigger than 3,000 Dalton, which was used to see how it modulates the gut microbiome and the metabolites it produces. Again, we're going to evaluate 4 of these mechanisms of hypertension: oxidative stress and vascular inflammation, and the renin-angiotensin system with the digested soluble fraction, and the gut microbiome with the digestion-resistant fraction. We first evaluated if the hydrolysate has any capacity to reduce oxidative stress in gastrointestinal epithelial cells. Here, we induce the cells to oxidative stress with a chemical inducer, and with a fluorophore, we detect that is activated by reactive oxygen species. We detect the fluorescent signal that was correlated to the amount of oxidative stress in the cells. The cells were treated with quercetin as a control and the hydrolysate at different concentrations. As you can see in the panel A, the egg white hydrolysate was able to reduce the area under the curve of the kinetic analysis at 2 different concentrations, meaning that it was able to reduce oxidative stress in the cells. Now, in panel B, when we analyze how much of this oxidative stress was reduced, we saw that in 500 microgram per ml, up to 27% of the oxidative stress was reduced by the hydrolysate. Next, we evaluated the anti-inflammatory activity of the hydrolysate in endothelial cells, specifically HUVEC cells. Here, we pre-treated the cells with the egg white hydrolysate and the egg white soluble fraction for 2 hours. After 2 hours, we induced the cells into an inflammation state using TNF-alpha for 6 hours. After 6 hours, we took the cell media and we fixed the cells to analyze the cell adhesion molecule VCAM1 through InCell Western. Here we observe in panel 8 that the egg white hydrolysate was able to reduce the expression of VCAM1 at 2 different concentrations. In contrast, when we used the egg white protein soluble fraction, there was no decrease in the expression of VCAM1. And this can be related to the peptide profile that we have in the hydrolysate, which could have peptides with a structure that enables them to exert this anti-inflammatory bioactivity. So now we have confirmed that the hydrolysate can reduce oxidative stress and inflammation in endothelial cells. But what about the gut microbiome? To evaluate the impact on the gut microbiome, we prepare a fecal slurry using the gut microbiota of 1 healthy patient and mix with the egg white or the egg white hydrolysate digestion-resistant fraction. We also use peptone as a control and perform an anaerobic in vitro microbial fermentation at different time points. We characterize the pH, short-chain and branched-chain fatty acids in the ammonia in the fermentation media, and we also perform 16S RNA sequencing to evaluate the composition of the gut microbiome. We observed that the pH was drastically reduced for the fermentation that contained the egg white or the egg white hydrolysate up to a pH of 5 compared to the peptone. And this is important because at a pH below 6.5, the Clostridia dominates the fermentation. Now, when we evaluated the composition of the gut microbiome, here we have in the red box the initial inoculum or the initial composition of the microbiome. In the yellow box, we have the fermentation with the egg white. In the blue box, we have the fermentation with the egg white hydrolysate, and in purple with the peptone. As you can see over time, there is a dynamic change of the gut microbiome composition, in which after 6 hours in the egg white hydrolysate group, there is a increase of the Bacteroidota and Actinobacteriota phylum. Now, as time progresses, the Bacteroidota phylum decreases as well, with an increase of the Proteobacteria and the Actinobacteria. And this is important because this composition will be responsible for the metabolites that we'll observe in my next slide. I'm going to start talking about the positive metabolites such as the short-chain fatty acids. And here we observe that butyrate and acetate were present in a higher concentration in the fermentation media when it contained peptone compared to the egg white or the egg white hydrolysate. However, for propionate, the concentration of propionate was higher in the sample in the fermentation treated with the egg white hydrolysate at 6 hours compared to the egg white or the peptone. And this is important because propionate has shown to be anti-inflammatory and as well to help reduce the blood pressure in pre-hypertensive patients. Now, when we evaluated the detrimental metabolites such as ammonia or the branched-chain fatty acids isobutyrate and isovaleric, we saw that these metabolites were in a higher concentration for the fermentations treated with the peptone. And this is promising because we can see that with the egg white hydrolysate digestion resistant fraction, we get more positive metabolites as propionate with a lower concentration of detrimental metabolites characteristic of proteogenic fermentations. We also validated the anti-hypertensive activity of the hydrolysate in vivo using a hypertensive rat model such as the spontaneously hypertensive rat. This model has a hyperactive RAS pathway which develops hypertension over time. We also have a normotensive control such as the Wistar-Killer rats. Each model had its own control and a group treated with the egg white hydrolysate at 1 gram per kilogram of body weight. And after 12 weeks of treating them with the egg white hydrolysate, we observed that the systolic blood pressure and the mean arterial pressure was lower for the spontaneously hypertensive rats treated with the egg white compared to its control. We also observed that there was not any change in the blood pressure of the Wistar-Killer rats, meaning that the hydrolysate does not affect the blood pressure in a normotensive model. The relevance of this study relies in that it validates the full hydrolysate as an alternative to single target drugs to reduce hypertension, and as well, it lays the groundwork to develop new type of preventive alternatives to reduce hypertension progression. I would like to take a moment to thank my advisor, Costa Majumder, my collaborators, and my lab mates as well for all of their support. Thank you, and I'm happy to take any questions.
Thank you so much, Emerson, for sharing your work. We do have a question in the chat. Someone is asking, which strains were used for fermentation?
So here we didn't use a specific strain. We used we prepare a fecal slurry by taking the stool sample of a healthy patient. So we will have a complex microbiome as well. We are in the process as well of including more microbiomes from different patients to see how is the variability of person to person as well, and evaluate if the results hold in the microbiome of different patients.
You mentioned simulating GI digestion. Can you expand on that and like how you do that?
Yes, so we use a standardized model called the InfoGest model. So in this protocol, we use a bioreactor in which we keep the pH constant For example, in the gastric phase, we keep the pH constant at 3 using the enzyme pepsin for 2 hours. And then we continue with the intestinal digestion using the enzyme pancreatin for 2 hours. This is an in vitro model. It is not intended to evaluate the dynamic change of the hydrolysis of the protein throughout time, but it is very comparable to in the endpoint results that we get from in vivo models. So in that case, we can evaluate what happens at the end of the digestion once we digest the sample.
Very interesting. Thank you. We have one more question. Do you think there's a role to evaluate different doses of egg white hydrolysate? Could there be increased BP control?
In the animal study, we set the 1 gram per kilogram of body weight dose as the maximum limit because it represents, by using an equation, it represents in the human dose, it represents that 20% of the protein intake for a human, which is kind of the limit that we want to set for example, nutraceutical food or functional food. But it has, as it has been observed as well, we have a significant decrease in the blood pressure up to 30 millimeters of mercury, which is often very good in an animal study. Of course, we will need to evaluate these results in a future as well in a clinical study to see if this sharp decrease in the blood pressure as well is observed.
Great. Well, thank you so much, Emerson, again, for sharing your work. I will now introduce our next speaker, Dr. Prasenjit Saha. So Dr. Saha conducted his doctoral research at the Indian Institute of Science in Bangalore, India, focusing on rare mitochondrial diseases associated with heart failure and neurodegenerative disorders. Currently, he works as a staff research scientist at the Cleveland Clinic in Ohio, studying the impact of the gut microbiome on cardiovascular disease. His research aims to identify novel cellular target receptors for human gut microbe-derived metabolites that are pathologically linked to CVD, with the potential to be used as therapeutic targets. So with that, I'd like to go ahead and hand it off to Dr. Saha. Thank you so much for being here and for sharing your work with us.
Okay, thank you for the kind introduction. So as the title of my talk is, The Way to My Heart is Through Beta-2-Adrenergic Receptor, Crosstalk between the gut microbial metabolite phenylacetylglutamine and the receptor. So as we all know that our diet contributes to cardiovascular disease. And our gut microbiome plays an important role in this because gut microbiome, it produces gut microbial derived metabolites that directly or indirectly contribute to cardiovascular disease. So in a nutshell, what we do in our lab is that we try to identify all the gut microbial derived metabolites that are associated with cardiovascular disease. One of the major metabolites that has been identified in our lab is trimethylamine N oxide, or TMAO. A couple of the last few speakers talked about this metabolite. And one of the very recently identified metabolites in our lab is phenylacetylglutamine, or PAG. In today's talk, I'm going to talk about this compound, phenylacetylglutamine. For the rest of my talk, I'm going to refer this compound as PAG. So what is PAG? PAG, excuse me. So PAG is a gut microbial derived metabolite that tracks with cardiovascular disease, heart attack, stroke, and death. So a dietary phenylalanine that converts to phenylacetic acid by gut microbiome, and that goes into the liver, conjugates with glutamine, and forms this compound phenylacetylglutamine. Now this compound PAG can act on resting platelets and make them hyperresponsive, and that leads to in vivo thrombosis, and that leads to thrombus formation and ultimately contributes to heart attack, stroke, and death. So my project is to identify the receptor of this compound and to understand the downstream signaling and how it contributes to CVD. So the first question that I asked, so does PAG binds to a cell surface receptor? So for that, first we try to see if PAG is binding with GPCRs. So GPCRs are the largest family of membrane receptors and targets almost 35% of the prescribed drugs. So if we see the GPCR signaling here that when a compound binds to GPCR, it can go majorly in 3 different pathways. One is G-alpha S subunit pathway, the second one is G-alpha I pathway, the third one is G-alpha Q pathway. And if this G-alpha S subunit is activated, that leads to production of cyclic AMP. If G-alpha I is activated, it leads to reduction in cyclic AMP. In case of G-alpha Q, we do see increased level of calcium. So first we check that does PAG increase cyclic AMP? So if we see here that in this graph that with PAG, we do see that there is a significant increase in cyclic AMP level. And when we block this pathway using a cholera toxin, we could see here that there is a significant reduction in the cyclic AMP production. However, in case of G-alpha I and G-alpha Q subunit inhibitors, we don't see any significant reduction. Also, when we used a global inhibitor of these pathway, which is called SCH, we do see a significant reduction in the cyclic AMP production. So the bottom line here is that PAG mediates cellular response through GPCRs via G-alpha S pathway. So now the question is that we know that it's signaling through GPCRs, and there are almost 800 genes present in human GPCR genes. So the question is that which GPCRs out of these 800 GPCRs? So at this point of time, the project was like this, like finding cats in this picture. So it was very difficult. So we took the hard way. So we tried to identify, we just took a silencer select human GPCR siRNA library, which consists of 379 unique GPCRs. And we tried to screen GPCRs in this pool. And after the screening, we found out that PAG is functioning through majorly 3 receptors: alpha-2A adrenergic receptor, alpha-2B adrenergic receptor, and beta-2 adrenergic receptor. So alpha-2A and alpha-2B regulates blood pressure, and beta-2 it regulates cardiac contractility. So since as I already said that these are the receptor alpha-2A, alpha-2B, and beta-2, so we used a pharmacological inhibitor of these receptors. So we used RX for inhibiting alpha-2A and alpha-2B, and used propranolol or carvedilol to block beta-2. And then we checked for platelet aggregation or also in vivo thrombosis. So as you could see here in this graph that with PAG, we do see a significant increase in human platelet aggregation. But when we used this propranolol or alpha-2 inhibitor RX, we do see there is a significant reduction in platelet aggregation. Similarly, when we did in vivo thrombosis, so we checked thrombosis in carotid artery. So that picture that you see here, that's the carotid artery, and the occlusion is seen here. It's the formation of thrombus. So if we see that in case of saline control or carvedilol control, we do see the formation of occlusion or the formation of thrombus within like 12 minutes. However, in case of PAG, there is like really early formation of thrombus. So we do see a thrombus formation in 8 minutes. However, in case when we used a beta blocker, carvedilol, so we do see that it comes back to normal aggregation time. So these experiments suggest that alpha beta blockers prevent PAG-induced platelet aggregation and in vivo thrombosis. So the cats were found at this point in time. So we found at least 3 receptors are there that PAG can interact. So now, at this point of time, we published this paper into 2020, which shows that PAG functions through alpha-2A, alpha-2B, and beta-2 adrenergic receptors. So now when I say that PAG acts via adrenergic receptor, so what does exactly mean by the word acts? So does it functioning as an agonist or it's functioning as an allosteric modulator? So before that, I'll just briefly describe about agonist and allosteric modulator. So agonist binds to the orthosteric site of a receptor and it can trigger a direct biological response. Whereas if we see the dose response graph of an agonist, you could see it could be like a full agonist or it could be a partial agonist. Majorly, these 2 are the classes. Now, an allosteric modulator binds to a topological different site in the receptor. It can either change the affinity of the agonist binding or it could change the efficacy of the biological response by an agonist. And if you see a graph of an allosteric modulator, you could see that the dose response of an agonist in green line, it can move to the right side or it can go to the left side, or it also can go to the up or it can go down. So if there is change in EC50 or change in affinity, we do see a movement in the right or left. If there is change in efficacy, we see it goes up or it goes down. So with this background, we wanted to know is PAG functioning as an allosteric modulator or an agonist. So we could see here in this first one that PAG is functioning as a negative allosteric modulator. So you could see here that in a competition binding experiment, we do see that PAG in black line is not able to like displace an agonist, suggesting that it's not binding to an orthostatic site, it's binding to a different site. And also you could see the dose response graph here that if we compare with this one on the top right, so it's moving the dose response of an agonist to the right side. Similarly, in case of like a cyclic AMP response, we do see that it's moving the dose response towards the right side, suggesting that it's functioning as an alpha negative allosteric modulator. You also take another biological response, which is called beta-arrestin 2 recruitment. In that case, we do see there is a significant reduction in the Bmax, suggesting that it's lowering down the dose response of an agonist. So the summary here is that PAG is a negative allosteric modulator. Now, the obvious question comes that how PAG functioning is as a negative allosteric modulator is physiologically relevant. So if you see in heart cardiomyocytes, there is beta-2 adrenergic receptors. And when a beta agonist binds to a beta-2 adrenergic receptor, it increases cyclic AMP that results in increased cardiomyocyte contractility. And there is that results in increased inotropic response. Now, in our scenario, we see that in presence of PAG, there is decreased level of cyclic AMP production. So the obvious question that does is also reduces the cardiomyocyte contractility. So here we did with mouse cardiomyocyte, we did the experiment, we checked contractility. So if you see here, this is beating cardiomyocytes here, and as it beats, it forms a cardiomyocyte transients. So in this graph, we could see that as in presence of an beta agonist called isoproterenol, there is a significant contraction. The more this thing goes down, it suggests that there is more contraction. So in presence of isoproterenol, we do see a significant contraction, but whenever there is a presence of PAG, this contraction reduces. So when we plot this as bar graph, we could see there is almost like 35% reduction in the cardiomyocyte contractility, suggesting PAG negatively regulates ex vivo cardiomyocyte contractility. The similar experiment we did in human cardiac muscles, failing human hearts. These are the patient samples that comes to Cleveland Clinic, and we obtained these failing heart tissue samples. And then we did the similar experiment. So we checked contraction in the heart muscle using norepinephrine. So we do see that with increasing concentration of norepinephrine, there is a significant developed tension in the heart muscle. However, there is a presence of PAG. We do see there is a shift of this dose response towards the right side, suggesting that PAG functions as a negative alpha negative allosteric modulator in case of ex vivo isometric cardiac muscle contraction. So now also we wanted to know where exactly PAG can bind in this beta-2 adrenergic receptor. So for that, we did some in silico analysis, and we came up with 5 candidate binding sites that PAG can probably bind to this beta-2 adrenergic receptors. So those are the 2 are at the extracellular binding domain binding site, and 3 were like internal, which we named as allosteric site 1, allosteric site 2, and allosteric site 3. And based on our in silico analysis, we found out what are the residues that could be involved in these sites. So we made mutation in these residues, and we checked for PAG allosteric activity. So I just don't want to go into like more details. I just want to like show the positive result we got out of all these sites. So here we could see that in case of wild type, there is a significant shift of this dose response towards the right side, but only in case of this allosteric site 1 mutants, we don't see a shift of this dose response, suggesting that these mutants, E122L and V206M, these residues are critical for PAG's allosteric function. So with this, I just want to come to the summary of my talk today. So in summary, we could say that our dietary phenylalanine, PAG produces from the dietary phenylalanine, and it binds to an allosteric site of beta-2 adrenergic receptor. And once it binds to the allosteric site, it reduces a beta agonist binding that reduces cyclic AMP production and also beta-arrestin 2 recruitment. That results in a decreased cardiomyocyte contractility. That results in reduced heart contractility, and that contributes to cardiovascular disease and heart failure. And also we showed that in case of this beta-2 adrenergic receptor, E122 and V206 are the critical residues that are important for the PAG allosteric activity. So with this, and also I just wanted to say that this study reports PAG as an endogenous allosteric modulator of a host GPCR for the first time. So with this, I would like to thank you all, and I'd like happy to take a few questions.
Great. Thank you so much, Dr. Saha. That was a wonderful talk. We do have a question. Someone is asking, could you give examples of the foods where we end up having TMAO and PAG?
Yes, it's just like mostly what that we are eating. It's like mostly the protein diet. So if it is like rich in protein, so that would like end up like producing more of TMA or PAG. So like the meat, like red meat has like mostly it just like contributes to production of TMAO. And for as far as PAG is concerned, so we are just at this point of time saying that more protein-rich diet, like I like hate to say that, but more protein-rich diet can like lead to production of PAG.
So someone else is asking, does PAG have any activity against other beta adrenergic receptors?
Yes, that's a good question. So yes, so there are like 9 adrenergic receptors, and we checked in all 9 of them. So at this point of time, we know that it's going through alpha-2A, alpha-2B, and beta-adrenergic receptors. So 3 receptors at least we know at this point of time. And other than that, maybe might be some receptors, but we don't know at this point of time.
Also, so I have a question. I noticed that when you were inhibiting for alpha-2A and alpha-2B, it seemed there was one inhibitor to block both receptors. Are there
Yes.
inhibitors available that can block one or the other, or are they too similar in structure and function for
Yes,
that?
that's a good. So we used a non-selective inhibitor just to like make our life simpler, but we do have like specific inhibitor for each of these receptors. So we use those as well. So just for like the time limitation, I did not show all of them. So we do have like specific inhibitor of like each of these receptors.
Gotcha. All right, thank you. Thank you so much, Dr. Saha, again. So it is my pleasure to introduce our last speaker for today, Sachin Aryal. So Sachin graduated with a master's in biomedical sciences from the University of Toledo in 2021. He is currently a PhD student at the University of Toledo in the Department of Physiology and Pharmacology. His research is mainly focused on the role of gut microbiota epigenetics in the regulation of blood pressure. He is a member of the American Heart Association and a peer reviewer for various journals such as Scientific Reports, BMC Bioinformatics, Biochemical Genetics, Phenomics, and BMC Genomic Data. So with that, I'll go ahead and pass it off to Sachin. Thank you so much for being here and for sharing your work with us.
Thank you so much for that kind introduction. Let me just share my screen. You guys can hear me, right? Yes. So,
Yes.
sorry, let me just, yeah. So, good morning, everyone. I'm Sachin Aryal. I'm a PhD candidate in the Department of Physiology and Pharmacology, and I would like to thank Proteintech Group for this opportunity where we can share our research work. And the topic of my talk today is on combating hypertension beyond genome-wide association studies, microbiome and machine learning as opportunities for diagnosis and treatment. So before going into my presentation, I would like to say that I don't have one particular research that we have been seeing from other speakers, but I have I have like multiple projects that I have tried to integrate to come together for the conclusion I'm trying to share with you guys today. So as we all know that cardiovascular disease is the world's #1 killer, and the stats over here, we can see that 33% of all global deaths are because of the cardiovascular diseases, and it is a group of disorder that affects the heart or the blood vessels, and hypertension or high blood pressure is one of the leading risk factor for cardiovascular disease that causes over 10 million deaths worldwide each year. And it is also called as a silent killer because it can lead to multiple other diseases and also kidney failure. So it is very important to give more attention to cardiovascular disease because it is one of the #1 killer. Currently, there are various diagnostic methods in use, such as just X-ray or electrocardiogram, cardiac catheterization, and many more. But even after all these advancements in this medical field, we can still, cardiovascular disease is one of the #1 reasons for the worldwide mortality. So systemic screening of any cardiovascular dysfunction can definitely save diagnostic time and initiate early therapeutic interventions. Sorry. So how about using microbiome and artificial intelligence as new opportunities? So this trend graph here basically refers to the number of publications in PubMed from 2000 to 2022, where we can see that microbiota and artificial intelligence related hypertension studies have been going up, throughout these years. And even in rodent studies, like in animal studies, we can see that microbiota studies are gaining much more popularity. That means microbiota and artificial intelligence are actually, they are being used in hypertension related studies and research work. So gut microbiota, as our prior speakers, they have they have talked about the gut microbiota and its role in various kinds of cardiovascular diseases. But just to summarize whatever they have mentioned already, so gut microorganisms, there are trillions of microorganisms in our body. Some of them are good, some of them are bad. Some of them turns bad because of the different conditions, different diet conditions or environmental conditions, and yes, so as we can see here that various of our activities like probiotic intake or dietary fiber intake, it results in the healthy conditions of the body, and then the microorganisms are responsible for a variety of functions like increased short chain fatty acid production or low in gut inflammation, and that results in the healthy condition. But when there is like high saturated fatty acid intake or sugar intake, excessive protein consumption, then there is the decrease in the short chain fatty acids or TMA production is increased, and this results in the disease condition. So gut microbiota has variety of roles depending on the conditions that we create. So based on that microbiota, when there is dysbiosis of microbiota, when there is a balance, when the balance between the good and bad bacteria is lost, or when this dysbiosis, it results in variety of disease conditions, as we can see here in this picture. And I would like to point out that in 2015, our lab was the first to report the evidence for a link between gut microbiota and hypertension in the Dalsal sensitive rat models. So before that also, and after this study was published, there have been multiple studies which have looked into gut microbiota and their role in hypertension. And yes, we have been gaining more information, with every research. So shifting gear to artificial intelligence or machine learning, as we have been hearing about, artificial intelligence, ChatGPT or GPT-3 and all these things that is ongoing right now in a rapid pace. There are machine learning and deep learning, which are the subsets of artificial intelligence. And these have been used in a variety of settings like business or education, gaming industries, a government. But my main focus is today in the healthcare setting. This excerpt from one of the papers I co-authored in, which is published in Journal of American Heart Association. It briefly summarizes the way how machine learning or which machine learning models or methods have been used for different clinical management of hypertension, or whether it be a personalized treatment. And there are many studies that are ongoing right now and that have been published where machine learning and artificial intelligence have been used for the management of hypertension. And since I talked about gut microbiota and I have talked about artificial intelligence and machine learning, so how about combining both gut microbiota and machine learning in order to predict the hypertension or cardiovascular disease conditions? So that's what my research was, which was published in Hypertension. So from the American Gut Project database, I got the metagenomic sequencing data sets. And from there, we got the bacterial OTUs data set, and that we used to train our machine learning model. And then the machine learning was able to predict if the people that sample that we used, from here, if they have cardiovascular disease or not, based on the bacterial OTUs. So when I did the analysis, found that there are a variety of microorganisms which are present in cardiovascular disease condition versus non-cardiovascular disease condition. So here we can see that, suppose, for example, Faecalibacterium is highly abundant in the non-cardiovascular disease condition compared to the cardiovascular disease condition. So this graph show that in non-cardiovascular disease condition, or the healthy condition, we can say that the diversity is higher compared to the cardiovascular disease condition. And we used those bacterial OTU features to train our machine learning model. And based on the random forest algorithm and the top 25 highly contributing OTU features, we got an AUC of 0.70 in differentiating cardiovascular disease patients versus non-cardiovascular disease patients. So AUC of 0.7 is good, yet not clinically acceptable. So my current research will, I'm looking into ways for increasing the AUC and other performance metrics of the machine learning, such as specificity, sensitivity, or other performance metrics, so that we can increase the accuracy even higher by using those metagenomic data and the machine learning. So our focus came to Faecalibacterium because it was highly abundant in non-cardiovascular disease condition compared to cardiovascular disease condition, and reports have shown that Faecalibacterium is it's a beneficial microorganism, so such a fatty acid producer, and its abundance is lower in disease condition compared to the non-disease condition. So we try to relate it with another study, which is chronic kidney disease conditions. So since we know that cardiovascular disease is one of the risk factors for chronic kidney disease, or vice versa, so we wanted to use Faecalibacterium to see if it can help in reducing any kind of kidney disease or not. So why was that necessary? Because chronic kidney disease, right now kidney disease are managed or treated by dialysis or kidney transplantation, but the limitation is that it's expensive and long hospital visits are necessary. Therefore, innovative strategies are needed for better clinical management of chronic kidney disease. So we hypothesized that if Faecalibacterium prasinujii can be used as a probiotic to lower chronic kidney disease and protect renal function. So what we saw, like as I previously mentioned, there was the decrease in the Faecalibacterium abundance in the CKD condition compared to the non-CKD condition. And this data set we used for the Western Hemisphere, we use American Gut Project database, whereas for the Eastern Hemisphere, we had our Chinese collaborators, and we had the data from the Chinese data set and also from the human samples. So we found that Faecalibacterium was significantly reduced in the non-CKD versus CKD conditions. And then we used a mouse model for the kidney disease mouse model, 5A6 nephrotomy model, where it was fed, where the mice was fed with Faecalibacterium prasinujii for like 3 times a week for 16 weeks. And altogether the study was for 20 weeks. And then we tried to see if feeding Faecalibacterium can actually lower kidney disease condition or not. And what we found was like serum creatinine and blood urea nitrogen were significantly reduced in the Faecalibacterium prasinujii treated group versus the vehicle controls. Also, we found that in this mesenteric staining, we can see that this blue stains in the CKD condition, we can see, like higher fibrotic area, but when there is a Faecalibacterium fed in those conditions, the fibrotic area is kind of, it's going away. So Faecalibacterium prasinujii was actually able to reverse the fibrosis, renal fibrosis as well. And more than that, also we looked into some of the renal inflammatory markers where we saw the similar result consistent with our previous one, that Faecalibacterium treated group had a significantly lowering of the inflammatory markers as well. So this brought to a potential translational impact that Faecalibacterium might be useful as a probiotic in order to lower the kidney fibrosis and inflammation and increase the kidney function. So when that can happen, we can also expect in some of the cases that it might be helpful for the lowering of the cardiovascular disease conditions as well. So this was our initial question: is microbiome and artificial intelligence are they as the new opportunity for the lowering for the clinical management of hypertension? And this picture is from our recently published review paper, where we propose that we can combine gut microbiome data with the genetic data and clinical data. That clinical data has been used with machine learning, genetic data has been used with machine learning or artificial intelligence for prediction of the hypertension. But what if we combine gut microbiome data together with clinical and genetic data and use artificial intelligence for the precision medicine for hypertension? So this is our proposal that we have recently proposed, and we are working on to see if we can actually achieve better results with this approach. So that brings to the conclusion of my talk, that microbiota have linked with the various cardiovascular diseases, as we heard from all of our previous speakers and from the research reports. And also artificial intelligence has been increasingly used to predict and treat hypertension. So integration of microbiome with clinical and genetic data and use of AI machine learning may help in devising personalized care for hypertension. So I'd like to acknowledge my mentor, Dr. Joe, and all my lab members, collaborators, and all the funding sources. And yes, thank you so much for listening to my talk, and I would be happy to take any questions.
Thank you so much, Sachin. That is very interesting. So we have a few questions. The first question that we have is, how much was the dose of Faecalibacterium P in mice?
So for that one, we used 2 × 10⁸ CFU per ml of Faecalibacterium, like every oral gavage. Like we did it 3 times a week. So every dose, every oral gavage had 2 × 10⁸ CFU per ml.
Great. Another question is, did your metagenomic data set involve 16S data or WGS for the machine learning in CBD?
Yes, it used 16S RNA sequencing data for the machine learning. Yes.
Okay, cool. I have.
Yes, and yeah, sorry.
No, go ahead. I was going to move on to the next question, but no, please add.
Sure. Yeah, I was looking in the chat and looking at the question. So yeah, you can go ahead. Yes.
Great. So one more question is, so with your CKD models, did you assess any pelvic pain measures?
Pelvic pain, no, we didn't measure the pelvic pain.
No pelvic pain.
No.
Okay. So one more question that we have is, did you perform serial measurements to determine the amount of Faecalibacterium P increased in your patients?
I quite didn't get that question. Serial measurements.
Sorry, you had said that you did the probiotic with Prasenjit. Did you do serial measurements to show that giving that probiotic was actually increasing the amount of that organism in their compositional microbiome?
So after, you mean to say like after oral gavaging, did we measure the amount of Faecalibacterium in the mice? So that, I don't think we did that. So we came up with the number like consistent with the previous research reports. 2 × 10⁸ was actually, was generally suggested for the optimal growth of optimal abundance of Faecalibacterium in the mice after feeding that particular dose. So we came up with that number, and then we tried with that. It worked for us. So I didn't think we did any serial measurements with that. Yeah.
Great. So we have one more question, which will be our last question for today. So someone's asking, do you have any ideas of the mechanism of benefit for the F. prasenjit? Sorry if I'm pronouncing that wrong.
Okay, so so I didn't, for the sake of time, I didn't include all the results here. So Faecalibacterium, as we know, it's a butyrate producer, and so butyrate is it's a Sachin fatty acid. So it helps. It is one of the it is the energy source for the colonocytes. So we looked into the mechanism, and we found that it is through the GPR43 axis, because GPR43 is one of the receptors for butyrate. So it acts via GPR43. And then the beneficial effect of Faecalibacterium is through that axis. So that's the mechanism that we have figured out. Yeah.
Great. All right. Well, thank you so much, Sachin. And thank you again to all our speakers for sharing their work and for their wonderful presentations. With the last couple minutes that we have, I just want to share a few product slides that might be interesting for our audience today. So as many of our speakers have mentioned, gut dysbiosis is oftentimes associated with a compromised intestinal barrier. So Proteintech does offer tight junction markers such as ZO1, occludin, and claudin-1 to visualize the integrity of gut epithelia. We offer both polyclonal and monoclonal versions, and our ZO1 and occludin antibodies are also available conjugated to our Coralite 488 or 594 dyes. So our Coralite dyes have unmatched brightness and photostability and are equivalent to Alexa Fluor in spectral profile. And all our unconjugated antibodies undergo stringent validation so that you can be confident that you are going to obtain reliable results. And by using our conjugated antibodies, you have a faster protocol because you eliminate the need for secondary antibody incubation, and it also allows for easier multiplexing. If you would like your primary to be conjugated, but one isn't available, you can now label them yourself with our flexible antibody labeling kits. So Flexible is a novel antibody labeling kit that uses an affinity linker to conjugate fluorochromes, enzymes, and molecules in any buffer condition in just 10 minutes. So this means that you can label your antibodies for IF or Western blot and eliminate the secondary incubation step. Because the antibodies are labeled prior to incubation with your samples, Flexible is suitable for same species multiplexing. So you don't have to worry anymore if your favorite antibodies are all from the same species. So with that, I just want to mention that as an attendee of today's webinar, you will get a link to request a free 20 microliter unconjugated antibody sample. You will also receive a certificate of attendance for our verified attendees and a recording of this webinar. And we do ask to please allow 1 week for this follow-up email to be sent. So with that, I just want to thank everyone for attending our Gut Microbiome ECR event today. If you do have any questions after this event is over, please feel free to email us at events@ptglab.com. If you liked today's event and you want to learn more about our future events, please do follow us on our socials because we post about our upcoming webinars and workshops. And so once again, thank you to all our speakers and thank you to the audience for attending. And I hope everyone has a great, wonderful day.
Thank you.
Thank you, everyone.
Thank you.